Welcome to our online microbiology journal club. This is our journal club for February, and we are discussing this paper, which is Analysis of Bacterial Genomes from an Evolution Experiment with Horizontal Gene Transfer Shows that Recombination Can Sometimes Overwhelm Selection. And you can see that I made my little note at the end of that. So I think the title is a really good preview of what's coming in this paper. There's a lot of data, and it's particularly timely because this is a paper that's based on some of the work coming out of Richard Lenski's long-term evolution experiment with E. coli, or LTEE. And I think the 30th anniversary of the LTEE is approaching the 24th, I want to say. It's basically in a couple of days. And so if you're interested in learning more about that, it's a really cool project. It's been running for 30 years. They're up beyond 60,000 generations or something, I think. I'm not even sure how many generations they're at now. It's some ludicrous number. And that is, in its own right, a fascinating experiment that has a whole raft of cool papers that go along with it. But this one in particular just came out recently. And so we're going to talk about this one today and how it investigates the impact of recombination and horizontal gene transfer on bacterial genome evolution. Before we get to that, I just want to do some quick introductions. My name is Laura Williams. I'm an assistant professor at Providence College in Providence, Rhode Island. And right next to me is Nicole. So Nicole, if you want to introduce yourself.
Yep. Hello, I'm Nicole Suctio, and I am a sessional instructor at the University of Northern British Columbia in Prince George, British Columbia, where I'm currently teaching a 88 to 90 student enrolled course on microbiology.
And that takes up a good portion of brain power and time and energy and so forth. So hopefully that's going well. And then next to Nicole is Elena.
Hi, my name is Elena Zykova, and I'm a bioinformatics scientist at Metagenome Bio in Waterloo, Canada. And I'm a microbial ecologist by training, but I'm starting to learn machine learning to find features in metagenomes and microbiome data.
Awesome. Thank you very much. And thanks to both of you for being here. So there's a lot to this paper. And in the hour that we've got, I don't think we're going to really be able to touch on everything. So we're going to try to cover some of the highlights from the paper. And I'm also going to say that, and this is probably true for my co-discussants here, but it was a pretty hectic week for me. So I read this paper, but I'm by no means going to pretend that I have mastered this paper down to every detail. So certainly if somebody ends up watching this and you note something or you have a thought about a topic that we're discussing, feel free to leave a comment, or you can tweet us too. We're using the hashtag MicroJC on Twitter. So that's always, if you tweet something to that hashtag, I'll see it for sure. So if you've got thoughts on that. So to start with, I think it's good to kind of get oriented about what are we doing. So I liked the fact that this paper essentially goes back and reanalyzes some samples from a previous experiment that sought to address this question. So the key question here, and they've got a sentence, I'm just going to read it straight from the paper. In this paper, we revisit an evolution experiment in which sexual recombination did not appear to increase the efficiency of natural selection, nor did it speed up adaptation, contrary to some hypotheses about the evolutionary advantages associated with sex. So, oh, hey, there's Sangeeta.
Hi, I'll join you guys in a minute.
Okay. So what they did here is they took this long-term evolution experiment that was established from, this is like a long time ago. So they took E. coli and they established 12 E. coli populations. And then they've been passaging those E. coli populations in the same conditions over and over and over and over and over. And that is what is arriving at its 30-year anniversary. What they did with this paper, actually what they did in the previous experiment, is they took those populations that had evolved for 7,000 generations And they said, okay, we're going to take those populations and we're actually going to do an experiment outside of the long-term evolution experiment, outside of the LTEE. And we're going to look at what happens if you introduce bacterial donor strains that are capable of conjugation into these populations. So the donor strains can't grow on their own. They have auxotrophic mutations that mean that they can't actually grow unless you supply nutrients to them. But they do have an F plasmid, which is part of what forms the conjugative pilus. So these donor strains that they combine with the recipients are essentially supplying some additional genetic material that could potentially get recombined into the genomes of these populations. And their notion here is, when they first did this, is that they were going to explore the impact on adaptation when you increase genetic variation. Does that make sense so far? Okay, yeah. All right. So did anybody have any thoughts or questions about the samples that they were using, or the populations that they were using, or the LTEE itself, before we kind of launch into what they did and how they used that experiment?
I'm just going to go. And I think it's in the methods that their regimen of sort of promoting these HFR gene transfers was that they were exposing the 4 HFR strains, or the 4 auxotrophic strains with the F plasmid, to the LTEE population every 5 days. I just wanted to be sure that that was kind of what was going on, is that they did this repeated set of cycles of exposing them to the opportunity to have conjugation happen.
Yeah, that's that's my understanding as well.
Okay.
And I think you've made a key point, that they are using 4 different donor strains. So they have, so it says on day 0 and every 5th day thereafter, a mixture of 4 K through 12 HFR donor strains. So these are E. coli strains that have the F plasmid. The difference between the 4 is that the F plasmid is incorporated into a different place in the chromosome in each of the 4. And then they add that to the populations and allow them to conjugate for 1 hour without shaking. And then they would shake them, disrupt the conjugation. And those donor strains, I believe, are expected to die out because they can't, they're in a nutrient limited medium and they're auxotrophic. Good? Okay. All right. So when they did this,
Sorry,
no, go ahead.
I just, one thing I was not very clear on was, so they did this experiment one time, and then this paper that we are reading is a kind of a reanalysis of the results. So did they resequence the genome, which was not sequenced at the time, in 1997, when the experiment was first carried out?
Yes, that's correct. So when they first did this experiment, they only had access to a few genetic markers.
Mm-hmm.
And so they assessed those, I think they said maybe 10. So they had a handful of genetic markers that they could use to test these questions. But what they're able to do now with, genome sequencing, I mean, the strides that we've made since 1997 in terms of sequencing, they can now just go back and just do whole genome sequencing. So they're not limited to the 10 markers. They can actually look across the entire genome now and try to get a sense of genome-wide impact on evolution.
Yeah, okay. So did they do the experiment again?
No.
Okay, just sequence those populations again. Okay.
Correct.
Okay.
What they did do, and we will talk about this, hopefully we'll have time to talk about this, they did have a question about the population dynamics.
Mm-hmm.
They wanted to know if some of the mutations that they were detecting would persist and go to fixation, or if they were transient and they just happened to catch them at a particular frequency in the population just because of dynamics, population dynamics. So they do to my knowledge, the main experimental work that they do here is that they actually take the populations at the end of the recombination experiments. So they basically grow those back up out of freezer stock and passage those a few times on their own. And then they look to see if those mutations that they detected are still there. Now, I don't know. I think that they did that specifically for this paper. It's called the continuation experiment. And I think that work was done. I think that work was done for this paper. I don't think that was done previously. Any other questions about this before we, excuse me, move on? Okay. All right. So one of the things that I really appreciated about this paper is, and as an instructor of microbiology and a college professor who's helping undergraduates learn how to read papers, I really appreciate that they were very clear about what their hypotheses were. They did not bury them in the text. It was not like when you're telling students, it's in there, you just have to find it. They're like, hypothesis 1 is blank. Hypothesis 2 is blank. You're like, thank you, that's so nice. And so what they essentially did was they said, we'd done this experiment. We had this handful of markers. Based on that data, we were kind of surprised because it essentially says that they found, you know, the stated goal of the original experiment was to test the hypothesis that recombination would increase the rate of adaptation by increasing the genetic variation available to natural selection. So that kind of makes sense. That's a standard evolution, molecular evolution hypothesis, that the more genetic variation you have, the more selection has to act on, perhaps you're going to see an increase in the rate of adaptation. But then they were surprised by what they found because they essentially discovered that this was not the case. So the outcome of the original experiment was unexpected in that the recombination treatment increased genetic variation, so that's expected, but it had no significant effect on the rate of adaptation compared to control populations that evolved without recombination. So that bit wasn't expected, and they weren't really sure what happened. So off that experiment, they had a couple of hypotheses that are like, well, maybe recombination acts like a strong mutational force. And so it's going to replace some neutral alleles because of the amount of donor DNA that's moving into the recipients. Or maybe some donor alleles hitchhiked into high frequency alongside some beneficial mutations that didn't really have anything to do with the donor DNA, but they just happened to be close enough that they got swept to fixation. Or maybe there's some kind of frequency-dependent selection going on. At the time, they really didn't have a lot of options for addressing these. And so now that they can do this sequencing, the whole genome sequencing, they can essentially go back and say, okay, well, what happened? What actually happened across the genome? Can we use that data to try to address what exactly occurred during this experiment? Good? Okay. So I'm going to move to the first, and I'm going to throw this up here. Let me see if I can share my screen. So I'm going to move to the first figure of the paper, which I cannot see. There it is. And I'm just going to put it up here because I think that this is useful to kind of go through. But I'm going to have to admit that I looked at this and I kind of went, there's a lot going on here. All right, so what we've got are 12 experimental populations, and those are the rows. And in this case, they're looking at particular clones from those populations. And then what they're trying to do, and you guys can tell me if this jives with how you guys interpreted this, but basically what they're showing is what was derived from the donor versus what was derived from the recipient. So what they're looking at here is, of these clones that experienced the recombination treatment, what proportion of the genome seems to have come from the K through 12 donor that had the F plasmid in its chromosome versus how much did they kind of retain of their own genome? And then eventually they also look at what happened over the course of the experiment. So to me, I think the key thing coming out of this is looking at the yellow. because the yellow is essentially coming from the donor. And they make a particular note that this one, that this is just basically an entire stripe of yellow. They're like, wow, that's a lot of donor DNA. I wonder what happened. And then they kind of stop there. I don't want to keep going on and on about this. What did you guys pull out from this figure? Does what I said kind of consistent with your read of it too?
Yeah, so I actually had a hard time interpreting this figure, to be honest. I did focus on the yellow because that was the only obvious thing to me. And I think there's something about the position of the genes that they're highlighting, but I wasn't sure. So I had a hard time with this figure, to be honest.
I did too, because I think it has a lot of moving parts. And I kind of kept going back and forth between the text and the legend and the figure itself. And I think the key thing coming out of it is to get a sense of, for me, I just kind of backed up and said, all right, so broadly, how for each of these populations, when we're looking at these clones, how much of the genome was actually impacted by recombination? And I think that for me, what it appears is that you're not seeing an extremely consistent trend. You're seeing a lot of heterogeneity in terms of how much donor DNA is showing up in each of these clones, which I think in and of itself is a really interesting point to emphasize, that, you know, recombination, there's a lot of chance going on here, as far as I can tell. Other thoughts on this?
I'm just going to go in and say that as a map to know what's happening, because I think if you look at sort of the window, or what's defined as sort of the x-axis on this, going to figure 2, which I'm not going to say too much about, at least it's sort of framing in on the area where within the amount of time that they gave the cells to hang out and for that pilus to form, it's the stuff that's most likely to be transferred. So that was helpful. I did start to get a little angry when I realized that there was also mutation information in here, and I'm just like, this is too much now. Like, this could have been blocked off somewhere else in another figure, especially when they're like, here are the different types of mutations, and I'm like, no, just stop. It's okay. But yeah, I totally agree with you that this is really interesting to think about the plurality of the extent of donor insertion into these recipient genomes. But I think it's also important to be mindful of the fact that this is also 7,000 generations of E. coli populations that have not really had that ecological context, you know, subjected to them in their history of that experiment. So, I mean, obviously this paper wasn't really designed to think about the physiological plurality and variability with now having this regimen of being exposed to HFR strains every 5 days. So I think thinking about some of the interplay that we don't know about that would probably be influencing what got transferred, what the length of actual physical conjugation events looked like if we could actually go in and like look at those cells in some sort of imaginary high resolution technique. There's a lot of diversity of the interaction, I think, that's being captured with a lot of factors that the paper doesn't consider, but it's worth reflecting on.
Yeah, and I would say for me, I feel like if someone did not have a really strong background in molecular evolution, there's layers to this paper that are going to be really hard to access. Like I would never in a million years try to teach this paper to an undergraduate student audience, ever. Like, I don't think they would, from the word go, they'd be like, what the hell is happening? Like, you know, because there's just a lot of, so you're trying to understand population dynamics, just the fact that these are clones of the population. So you have 12 experimental populations. The row that's showing up for each one of these is not necessarily what's going on throughout the entire population. That's a single clone. Now they do make a point of saying, like, we chose 2 clones, and throughout the paper they do point out, like, generally speaking, the trend that we're seeing here was also true for the other set of clones. But I mean, you've got population level dynamics, you've got the historical contingency of what happened in lineage 6 a long time ago, you've got selection, different forces of selection. I mean, my goodness, there's a lot going on. It has a lot of moving parts. I feel like, Nicole, you're about to say something.
Yeah, I feel you on that, because I did not choose academic trajectories that groomed me well enough in molecular evolution to even say that I'm remotely comfortable with sort of the discourse. So unpacking a lot of this it's actually really interesting. But I think this is also just really cool to think about, because again, they are starting with clonal E. coli populations. So the fact that you're getting this much diversity in something that really doesn't even, I guess, when you think about it from a genetic perspective, reflect like there's still diversity, even though these are genetic clonal populations, in terms of the interactions that probably took place, and in terms of maybe, again, those other contingencies of the HFR recipient interaction. So this is still really interesting to think about, because translationally, when we think about, risk and contingency and how the things we do influence selective forces, in more applied fields where we have to think about gene transfer and, say, anthropogenic impact, this is actually setting a really interesting stage for thinking about, well, there's still a lot of other outcomes that can happen even when you let a highly similar population do its thing, when this mechanism of genetic variation is at play.
Yeah, that's true. That's true. All right, does anybody, I'm going to move on from figure 1. Does anybody else want to mention anything about figure 1 before we go? Okay. So
I just want to ask you
one. Go ahead.
So the way they numbered these populations makes me think that, the first population is related to the 7th population somehow. It just threw me off for a minute there. So then I realized a little bit, is that a standard way of naming populations, or is there something they did?
And this is where I'm going to have to admit that there was a time when I actually had read some of the early papers from the long-term evolution experiment, and I probably could have answered your question about why they're named, like, I think this is the Arabinos thing, but +1 through +6, and then -1 through -6, but I can't for the life of me remember, and I didn't have time to look it up, but I was like, oh crap, I should really remember why they're named that way. And I honestly just don't remember.
Okay.
I think it's something particular to this experiment, and I just don't recall what it is at the moment. Nobody else ran across an explanation for that, did you guys? Okay. All right, so I'm going to move on to figure 2. So figure 2, they're kind of asking in another way. They're essentially thinking about, so they've got 4 donor strains. The F plasmid is incorporated into different positions in each of the donor strains. So the donor strains are, to my understanding, is that the donor strains are isogenic, except for there's an F plasmid sitting here or here or here or here. And so you've got, by virtue of how plasmid transfer occurs, see, again, this is another element for why there's no way I would ever teach this to undergraduates, because now you've got to spend forever talking about plasmid transfer, and they're like, what's happening? So basically, your plasmid that's been incorporated into the chromosome has an origin of transfer. That's where there's a whole bunch of proteins that interact with the origin of transfer. So once the conjugative pilus is formed, that's like the start of the tape, basically. So that's the bit that goes through first. And then everything that's in a particular order and orientation in relationship to the origin of transfer has a much greater chance of making it across the pilus to get transferred into the donor. This is like when you see for E. coli chromosomes, how we used to do it. They used to do it by minutes, because it was basically like when you tried to get the chromosome transferred across, if you split the transfer, if you cut off the transfer at like minute 30, how much had you gotten across? If you let it go to minute 45, how much more had you let go across? It's the same idea. So they basically, wanted to see where the origin of transfer is in each of these donor strains. How did that impact what got transferred into the recipients? And so that leads to this figure. Put that up there. All right. I still don't know where they got the introgression score from, and I was too lazy to go try to figure out if there, I'm sure there's like an equation. Maybe it's not that complicated, but I was like, I don't know where they're getting the introgression score from, and it's like 10:45, and I can't go look it up right now. So, but what they basically did here is, okay, no, their 4 donor strains are indicated by the 4 colors. So the arrows are showing you the origin of transfer and then which direction it's going to go. So what you've basically got is, as I understand it, you've essentially got the chromosome kind of laid out there. And then there's 2 peaks to this figure. And those peaks are kind of centered on the origins of transfer, and they're kind of defined by the directions of transfer. So what they're essentially saying is, because of where the plasmid is sitting in the chromosome in these donor strains, because of where the origin of transfer is sitting and the orientation that it's in, these regions that are showing up as peaks in the blue are going to be most likely to make it into your recombinant populations. You're going to see those. Those are the yellow things, basically. That's where the yellow is coming from. And they point out that there's a big gap kind of at each of the 2 ends. So if you're thinking about this as a circular chromosome, there's a big portion of the chromosome that just doesn't get represented in the recombinants that much. And they say, well, if you look at where the origins of transfer are, that kind of makes sense. Thoughts on this? Did anybody, they talked a little bit about how the auxotrophic mutations kind of act as barriers. I didn't really plan to go into a lot of detail of that, but I don't know if that's, does the organization of this figure make sense to folks?
It mostly does to me. I also don't know if I would dwell on the auxotrophic mutations being barriers to introgression, because I mean, it kind of, I'm okay mostly with their explanation of it. And I think they unpacked really well sort of like that temporal constraint on the conjugation event as sort of explaining a large part of the gap as well. You know, they do talk about the assumptions regarding how soon transfer is happening and the likelihood of those nearby regions of sequence to RET, pretty much sort of clarifying the way we expect a system like this to work for gene transfer. So I'm not like 100% like I understand everything, or even sort of the divots in the 2 peaks with respect to where things are showing up. But I'm mostly okay with it.
Okay. I think mostly okay. It's like, you know, it's like from Princess Bride. He's mostly dead. You could still get him back. So if we're mostly okay, we're going to go with it. Yeah, I don't think I'm going to dwell too much on this either, because I think the main things that come out of this is that there's a big stretch that doesn't show up as much, that didn't get transferred as much. And that seems to be a function of how they designed the donor strains. So in a way, that's a way of going, okay, well, given how we designed the donor strains, that makes sense, that you've got this portion that's showing up more than the others. All right. So they then start getting into their question of thinking about what actually happens as a result of that recombination. So based on the fact that we saw all that yellow in the first figure, And based on looking at figure 2, where they're actually showing these blue peaks, what that's indicating is that there is recombination going on in the recipient populations. The result of that is that you've got populations where you have a mix, the chromosome is a mix of recipient that was original to the recipient and then donor derived. So then they start going, all right, well, what actually happened in terms of replacement of alleles? So the next section that they've got, which is called probable beneficial mutations, is essentially them kind of laying out, all right, these are mutations that we think are under positive selection as a result of, or not as a result of, but they're under positive selection, and this suggests that they're involved in adaptation to the conditions of the long-term experiment. So remember, these are coming from this long-term evolution experiment that has been running for 7,000 generations. So each of these populations in that span of time is hypothesized to have accumulated some mutations that are involved in adapting to those conditions. And this, they've got table, which I'm not going to put up, but they've got table 1, which is essentially a list of 32 genes that they think are under positive selection that are having some impact on adaptation to the conditions of the long-term experiment that they're doing. Is that good?
Yeah, so this is, this is not really my wheelhouse, this molecular evolution, but I was a little bit confused, and maybe it's somewhere in the paper and I didn't see where, but I was confused as to how they were interpreted those 32 genes as being under positive selection.
Yeah, like how did they ID those as being positive selection? And I think that has to do with the G score. So if you look at the last column of table 1, there's a G score there. And it's a method of calculating the strength of evidence for positive selection. One of the ways you can do this in a, as I understand it, one of the ways you can do this in experimental evolution, this is why you have replicate populations, because if you see mutations accumulating, if you're applying the same conditions to all the lineages, and you see mutations accumulating in the same gene in multiple lineages, it's very unlikely that that's due to chance. And it's very likely that that's because that particular mutational pathway helps them adapt to those conditions. And that, I think that's one of the things that contributes to the G score, is how many mutations do you see in the gene, how many lineages, so how many replicate lineages are showing the same gene mutated, which gives you some extra confidence, okay, yeah, this gene probably really has something to do with adaptation, and it's not just it mutated by chance, and it's a neutral allele.
Yeah, that's really helpful. Thanks.
Okay. So we're currently running, it's on pause right now, I have to restart it, but we're currently overall doing an experimental evolution project. And we didn't do 12 because there's no way I could do it. So, but we've got 6 lineages going, and our justification was exactly the same. Like if we see mutations showing up in multiple of the populations, that's a little more convincing to us that it wasn't just a fluke, but it really does have something to do with adaptation. Okay, I think this section was a section like we're going to introduce this to you so that you guys know this, you have this list of genes in mind, and then we're going to come back to it kind of a thing. So I think the main outcome from that is, here's our table of 32 genes that we think are under positive selection, and therefore we're going to eventually later argue are beneficial. And we'll come back to this. And I think that's about all of you need to get out of this section for this point. So what I think we can do is we can move on to the next section, which is possible sources of variation in introgression across genomic regions and the fate of previously evolved beneficial mutations. Now, I have to admit, I had to read this section twice, in part because I read this paper in chunks, and when the session in which I got to this section, I was starting to get really tired. And I tried to read it because I was like, I need to get through this. And then I went to go back and start reading it again this morning, and I thought, oh, you didn't get anything out of that last time when you read that. I don't even know why you bothered. So here's what's going on. They're basically saying, okay, So we've got variation across the genome in how much recombination we actually saw. So if you go back and look at figure 1, that's one of the big things that leaps out of figure 1. It's not consistent. It's not like everybody's got a patch of recombination right here. It's like one clone was entirely donor-derived. One clone was barely donor-derived. Other clones had different patches in different places. So like, what the heck is going on? And so they present, again, I do appreciate this. They kind of present multiple hypotheses, which I basically summarize as it has nothing to do with selection. That's hypothesis, in my opinion, that was hypothesis 1. It's just that you've got some regions of donor DNA transferred more often than other regions, leading to overrepresentation. And to me, that really didn't really invoke much having to do with selection. And then hypothesis 2 was, hey, maybe there's some beneficial alleles in this donor DNA, so they're going to get selected for, and then they're going to show up more. And then hypothesis 3 was basically, well, maybe there's some donor DNA that actually has deleterious or disadvantageous alleles, so they're going to get selected against. So those are underrepresented. And then they've got some subparts for hypothesis 3. So that's kind of their structure for getting into this. And then they kind of went through and talked a little bit about evidence for one versus evidence for another. I don't know if I want to, there's a lot of text here, and it's a really interesting discussion of all of this. I'm not sure if I want to get into a ton of it, but did you guys have anything that you picked out of this that you're either like, I would like this explained, or I thought this part was interesting? I thought this was the trickiest section to just kind of like maintain, like, okay, I'm following along. I think I know what's going on, so forth and so on. They kind of walk through and stack up evidence for and against each of these hypotheses. So they end up basically saying, we don't actually think that there were a lot of beneficial alleles donated. So hypothesis 2, they're like, we don't really think that that's true. They find some evidence for hypothesis 1, that there's just like, because of that calls back to the second figure, where because of where the origin of transfer is located and the way it's oriented, you're just more likely to have certain regions that get transferred and then recombined. And then they talk about, there's some evidence that, as far as donor alleles being selected against, it certainly makes sense that if you transferred the oxytrophic mutation, that's probably going to get selected against because it's lethal. So it's unlikely that you're going to find that oxytrophic mutation in your recombinant populations. And then they kind of talked about, let's see. So they get down towards the end of this, kind of at the I think it's page like 10 of 30. So it's a part where they start discussing how new alleles kind of arise. And so they're essentially looking at, and I have to admit I kind of got lost here, because there's a couple of different like state changes that they start discussing. So you've got for a particular allele, you've got the donor allele, or the donor state. You've got the recipient state. You've got the ancestral state. And then it's something entirely new that we haven't seen before. And so they kind of start categorizing mutations as being like, all right, so there is an allele here on the chromosome. After the recombination treatment, does it look like the donor? Does it look like it was affected at all? Did it revert back to the ancestral state, or is it something entirely new? And so they kind of, in figure 3, they kind of just talk a little bit about 4 new alleles that they find. And to me, I think that I kind of looked at this and went, oh, that's really interesting, because it just kind of shows you where novelty in genes could come from in a natural setting. When you've got conjugation happening, when you have potential within gene recombination. So you end up with these kind of chimeric genes where part of it is from the donor, part of it is from the recipient, but it's still there and it's potentially still a functional gene. And that's kind of cool to think about, because then you're talking about introducing new protein domains and combinations that you maybe didn't have before and things like that. I saw Nicole was nodding. I don't know if anybody has thoughts on that.
I'll just go. I mean, I kind of lost trains of thought towards this section, but I mean, this is something that did stand out for me, just the fact that they did take the time to unpack a little bit of this. Here are some new sequence attributes of proteins that are definitely sort of showing up. But I think what's interesting to think about this even more is, like, if the continuation experiment was longer, to kind of look at some of these things that they flagged in terms of are they eventually turning into places with higher than expected mutation frequency, just because the diversity kind of introduced is already now there as a consequence of this HFR context that these lineages have been exposed to. And I think I can't really say anything in detail about this right now because I just did not absorb any of it, but it's definitely nice to see them address it sort of from the translational consequences that happen, particularly with that section D where you've got like the nonsense mutations sort of showing up. I mean, it's always kind of interesting to see that sort of pop in and then wonder how a cell's going to handle that.
I have to admit, one of the things I was thinking about, just because of stuff that I've been teaching in microbiology, one of the things I was thinking about with this is it just kind of also re-emphasizes, this is not news, this is something that people who do microbial ecology and microbial phylogenetics know very well. But just the notion that if you're trying to understand evolutionary relationships amongst bacteria and you're using a small handful of genes, if you've got like a hybrid gene, then you're like, I only got a half of it. Like, I mean, but that gets masked, right? Because you just don't, I mean, we don't think that way. We make gene alignments and then one half of our alignment looks like really bizarre and the other half looks really, and you're just like, I don't know. The tree just needs to make it do something. But it did make me kind of think about, this is really good data on why gene trees for bacteria can get so messy and why it's, you know, important to kind of have a pretty robust, you've either got multiple loci from different parts of the chromosome, or you've got, you know, whole regions and you're kind of scattering them around, or you're doing windows, or you're doing something. Because otherwise, if you just like picked one of these genes and built a tree, you'd be like, what the hell is going on with this tree? You know, it's unrelated to anything they were discussing for this, but it is something that I thought, yeah, that's like a good example about how quickly things can get in the weeds. And this is all stuff that's just really closely related to E. coli. So.
And it's interesting that there isn't really seem to be a pattern, whether it's more like going to be like recipient or more like the donor. And so going back to the point of trying to see this in like even closely related populations in environmental communities, you'd look at this chimeric looking gene and you wouldn't know which part of it was part of the original genome and which part of it came from somebody else. I mean, you'd start to look at other genomic features, but yeah, it's interesting to me that there didn't seem to be a strong pattern, whether it was mostly new things or it was mostly recipient or donor things. And I guess I would have liked maybe to see like a summary figure of that or something to see if there were patterns, because nothing jumped out at me. So I'm like, oh, so is this completely different for each one? Or I don't know.
Yeah, I agree. And I think that's part of, I think that's a through line throughout the whole paper is like, this didn't play out like we thought it was going to. What happened? And so I think that's part of, I think it seems to me just from reading this that the authors would agree, like, yeah, it's kind of odd that there's not like a predictable pattern coming out of this that we could tell. I don't know. I mean, Sangeeta, you're doing phylogenetics, yes? Is this something you have to contend with as well? Like thinking about how these genes recombine and how that impacts phylogenetic signatures?
Yeah, I don't do that kind of work myself a lot, but I have read papers where this kind of, it's maddening. And I'm working more on the mathematical side of things, where we are trying to unpack, if you see this kind of absurdity in the tree, which looks more like a network, could it have come from a network, or is it a network, or there's stuff like questions like that. So yeah, we have seen this, and we have discussed this in our like papers, etc.
Yeah, I mean, it seems like a big headache. It seems just, and especially if you're doing, I would think if you're trying to do stuff with metagenomics too, where unless you can get assemblies of genomes, you're working with chunks. And so then you're like, man, I don't know, is this a recombined chunk? Like what does this mean for the thing that it came from? Is it actually a valuable signal for that organism? Or did I just happen to accidentally pick something that's just incredibly recombined with other things? And so it's just like, nope, you had bad luck. But, okay, so given time, because I want to kind of hold to time, I'm gonna actually, there's a lot in this paper. Nicole and I were talking about, just before people joined us, Nicole and I were just noting that there's a bit in the materials and methods where the person who wrote I'm assuming this is the first author who put this note in there. It was like, there's an R script available to do all these analyses, and it's called dissertationanalysis.r. So this is like, I think this is a healthy chunk of the first author's dissertation. It's like, there it is. And you can tell because there's a lot in here. But I think what I'm going to do is I actually want to skip ahead for time, just because I want to look at, so they saw a lot of when they saw this recombination happening, one of the things that they were somewhat surprised by was the fact that it looked like beneficial mutations that arose during adaptation to the long-term evolution experiment conditions were getting replaced, that is very surprising. And so you would think, well, that's really odd, because there's no reason to think, they ran the conditions the same. So there's really no reason to think that those mutations are suddenly not adaptive to the conditions anymore. And there wasn't a whole lot of indication that the donor alleles were more adaptive or more beneficial. So they kind of ended up with a big puzzle, which gets back to the title of the paper. Recombination can sometimes overwhelm selection. So this notion that they're seeing donor-derived alleles that don't appear to have any fitness benefit, or have less of a fitness benefit than what was there before. And they were kind of confused by that. And they just basically confirmed, yeah, that's really what happened. We're not really sure why. But one of the things that they wanted to test was maybe, because a potential explanation is, well, maybe you just caught these populations at a point where things hadn't quite gone to fixation. So maybe you sampled these populations when a conjugative event and a recombination event had just happened. And so in this clone, what looks to you like a replacement of a beneficial allele is in fact one sample of the population genetics there, that if you let it keep going, that donor-derived allele would drop out, the beneficial allele would rise back up in frequency in the population, and maybe you're just kind of missing that. Maybe you caught it too early. And so what they did was they said, okay, that's fair. So they took their populations and they grew them in the conditions of the long-term evolution experiment, but they stopped doing any kind of recombination treatment. So they just passaged them and passaged them and passaged them so that they could get more time on the clock to kind of get the dynamics sorted out. So if the donor-derived allele was harmful, selection should have time to act, reduce it in the population, the beneficial allele should come back up to fixation, and then you'd see that reflected. Does that make sense? Okay. So what they did was they passaged it for a bunch of generations, and then they actually looked to find the frequency of the donor alleles. So I'm going to put up this figure because I kind of liked this figure just in the way it was organized. So this is figure 6. Basically what they're doing here is they're looking at a comparison of initial and final frequencies. So if you look at the legend, it's essentially saying, okay, so these are the K through 12, so these are the donor-derived alleles. And then what we've got, I think it's the x-axis is the initial frequency of the allele. So when they sampled it at the end of the recombination treatment, that's the frequency of the allele. And then the y-axis is the frequency after they passaged it a bunch of times without recombination. So after they kind of gave selection some time to act if it was going to. So if it's on the diagonal, if the allele is the dot is on the diagonal, then that means there was no change in frequency from initial to final. If the dot is above the diagonal, it means it actually increased in frequency from initial to final. And then if the dot is below the diagonal, it means it actually decreased in frequency from initial to final. So if your K through 12 donor-derived allele was going to be selected against, it would show up below the diagonal. If your K through 12 donor-derived allele is going to fixation in the population, it's going to show up above the diagonal. And I think, you know, there's a lot going on here. I mean, one of the things you note from this is that there's no real obvious clear pattern that everybody's showing, but you get things like this, error -2, where you've got this big blob of alleles that's showing up above the diagonal. So it's like, huh, so those donor-derived alleles are actually going to fixation, or they're increasing in frequency in the population over time. So even if they don't appear to have a fitness benefit, they're increasing in frequency. And I thought that was kind of interesting. I don't know how many people got it this far and still it still had the bandwidth to kind of process this, but I don't know if other people had thoughts about this figure.
Yeah, this figure is really, I mean, it's a little bit crazy that way. So even though there is no clear pattern, but you would see overall, if you wanted to summarize, you would see a lot more stuff above the diagonal than you would see below the diagonal. That's some pretty crazy result there.
Yeah, I agree with that. I don't think they really expected that either.
But I just wonder, so in the methods, they say that the ratio of donor to recipient cells during the treatment was 4:1. So I wonder
Okay.
if the ratio had been less, or like, you know, maybe 3:1 or 2:1, would that have made a lot of difference to the results? Because the donor is like 4 times as many donors as recipients.
Yeah, that's a really excellent point. I wonder if they picked that. I wonder if they picked that in the hopes that, because the donor, there's 4 donor strains, so there's a mix of 4 different donor strain types, I guess.
Yeah.
I wonder if they did that so that each donor strain type would be 1-to-1 with the recipient. I wonder if that was their thinking.
That actually makes sense, but then that is kind of overwhelming the donor, the recipient population, isn't it?
Yeah,
So
that's true. That's a really good point. I wonder how that impacted it. If they'd gone a 1-to-1 ratio, in effect, that would have been like for each donor strain type, it would have been like point, you know, you
25%.
Right, exactly. But that's true. I wonder how, and we kind of thought about this, or I kind of thought about this in setting up our own experimental evolution protocol. Because in our case, we're doing predatory bacteria and prey bacteria, and we're testing if predatory bacteria will adapt to prey, a particular prey species. And so for me, I spent a bunch of time, and I have like a scribbled notebook of lots of probably very bad attempts at working out the math of it, trying to figure out the ratio, you know, like how many predators should we add to how many prey, or should we be, what should the ratio be? And I do wonder how that kind of thing impacts an experiment. So does anybody have any other thoughts about this particular figure? Okay, let me do this. There we go. All right, so to me, I think they kind of, there's a couple other figures here. As I mentioned, there's a lot that's going on here. They also have a figure that looks at specific mutations that are at 100% frequency in both the initial and the final samples. And they look at the frequency of the F plasmid. So they have a lot of different questions they can ask about this. But I think kind of overall, what they're finding with this is essentially, and I'll just go back to their abstract as the best kind of summary of this. They essentially found, you know, beneficial alleles in the recipient populations were occasionally driven extinct by maladaptive donor-derived alleles, which is not what you would expect, but they've essentially confirmed the result that they had from the original experiment when they were working with just a handful of markers. If you expand out to the whole genome, you still see the same thing. And then they say, on balance, our analyses indicate that the plasmid-mediated recombination was sufficiently frequent to drive donor alleles to fixation without providing much, if any, selective advantage. And that's really interesting. And I don't know that we've got a great explanation for that, but that does work into thinking about recombination. And if you've got a situation where recombination is high, how do you factor that into thinking about potential impact on genome evolution? And I don't know that many models incorporate that kind of a factor. I don't know how many need to be perfectly honest, because this is a high rate of conjugation that I don't know what this matches to natural communities. So it could be that this is not nearly as big of a factor in natural communities, but I'm not sure. Thoughts on kind of the broad?
Yeah, so overall, like I'm looking at figure 6 too, like, and maybe my interpretation is completely off, but it seems like recombination can like interrupt some of these selective processes. And then over time, once the recombination stops, then certain genes get fixed and then other ones don't. But I think the recombination itself, because I mean, if you think about genome rearrangements, yeah, some new beneficial things are going to get in, but they're also going to break things that are in the genome that are already beneficial. And so maybe that's more a disruptive process rather than a selective process. And then you need time away from it to be selective. But again, this is not like my wheelhouse. So this is just what I got out of it. But I don't know if that's accurate at all.
Yeah, and I mean, I can imagine if you're thinking about microbial communities, this just kind of gets into, there is gene transfer that happens amongst microbes in communities, and so trying to understand like community dynamics and how to model, you know, the recombination part with selection. Yeah, this kind of shows that you know, recombination can be a factor in ways that we may not necessarily expect. Oh,
we tend to think about HGT in like microbial ecology as like, oh, they need this thing and they acquire it from this. But I mean, how much HGT is actually not necessarily beneficial to the cells that we see HGT in? And so maybe this should reframe the way we think about it when we find it in genomes. I don't know.
Yeah, yeah, you're absolutely right. And I mean, I definitely fall victim to that. I kind of teach it that way because you're trying to get students to understand it. So you talk about antibiotic resistance, and you're like, and then they get the genes, and then it's like, oh no. But you're right. You can't always assume. I mean, in some cases too, some of these mobile elements are selfish mobile elements, and they're just trying to replicate themselves. Some plasmids, they didn't get into this, and I don't know if it's a factor here, but some plasmids actually have their toxin-antitoxin systems, where it's basically, you know, if your cell divides and you don't get enough of the actual plasmid, the the toxin part kills the cell that didn't get the plasmid. So that's very
But
kind of.
this is a very artificial setup. So I don't know if we can necessarily propagate the conclusions from this experiment to natural communities, because conditions there are very, very different. So I think maybe, a decent mathematical model is probably needed to be able to transfer these learnings into my natural communities.
Yeah, that's a very fair point. This is a very artificial setup, and it may not translate perfectly into a natural community, especially a mixed community where you're going to have a lot more phylogenetic diversity represented in your microbial community. Nicole, did you have any kind of broad thoughts on this?
Yeah, I think I appreciate Elena's comments about disruption, because I mean, really, this is a story about vandalizing a genome with HFR strains. I mean, you just wrecked stuff close to the RET, and part of me, when I think about that diagonal figure with all the clumps, which I wish I could remember the number for, but I can't apparently. It's color coded by genome position, which they do say is haplotype. But I think the other thing to think about there is you vandalize the genome, but these things still want to stay alive. So if you're fixing mutations, I mean, part of that may also have to do with some slightly distal cis-acting elements. It might have to do with mutations that are actually allowing parts of regulatory systems to interact with that DNA to coordinate. There's pleiotropy of phenotypes. So like, how do you keep the whole chromosome doing the things it needs to do to keep the cells doing the things they do in the face of that type of variation? So I mean, there's that whole bucket of worms to kind of contend with. And I think that's what makes this also really interesting, is like, we get this genomic snapshot, but then you also have to think about, okay, we can't necessarily tease out, except by frequency, what's positively selected and what's leaving, but we also don't know all of the players that are trying to really promote homeostasis and function about those regions of DNA that have just kind of undergone this sort of flux.
Yeah, I think that's a good point too. So we've arrived at an hour. So I just want to. I was happy to read this. I think the timing was really good. Selfishly, it gives me some stuff to think about as we're working with our own experimental evolution project. So I thank everybody for indulging me in this one. It's a really interesting paper. There's a lot packed into it. And there was a lot of layers to kind of get through. So for those for whom this is maybe not the wheelhouse, thanks for kind of sticking with it, and a really good discussion too. So I do want to thank everybody for coming to discuss this paper. So the pattern for the spring is we're going to be last Thursdays of the month. So that means that we are looking at, let me get my calendar up really quick. We're looking at Thursday, the 29th of March at 3 p.m. And if anybody has, I know a couple of people, I think Sangisa, I think he suggested a paper before. You might have moved on from that paper, but there were a couple other paper suggestions. So if people do have paper suggestions, we tend to focus on microbiology papers, but if there's something that somebody wants to knock off their to-read list, I'm always happy for suggestions, and we'll put it on the list for sure. Does anybody have any final thoughts before we sign off? Okay, so with that, I want to thank everybody for showing up, and hopefully look forward to seeing you guys next time.